Alright, so yesterday we finished talking about the differences between the rough ER and the smooth ER. So the rough ER was more associated with modifying proteins, which made sense because the ribosomes that are found around the rough ER, they made the proteins. So it's kind of convenient that then the rough ER could modify those. And then the smooth ER was more associated with lipids, detoxifying drugs. It also could do some stuff with carbohydrates, but not proteins. So that's the difference between them. They both look kind of like a series of canals, so they sort of have that in common. They're also located, notice, very close to the nucleus. So it makes sense, that instructions from the nucleus are sort of carried out at the ribosomes building the proteins, and then they're modified, and now we're working our way closer to the cell membrane away from the nucleus. So our next organelle is called the Golgi apparatus. It's also sometimes called Golgi complex. I've also heard it pronounced Golgi instead of Golgi. So it looks something like this. Oh, and Golgi bodies is yet another name for it. Why it has so many names, I'm not really sure. It's like a stack of short membrane-like sacs. So there's a stack of sacs, kind of like a stack of pancakes. And what it does is it's really known for modifying and packaging molecules. So they're either going to get stored or they're going to be excreted from the cell altogether. For example, you being a multicellular organism, you have cells, for example, that their job is to make a particular hormone. And that hormone has to travel through your bloodstream to other cells to then carry out its function. So that protein hormone would be made by the ribosomes, modified by the rough ER, and then carried in a vesicle to the Golgi bodies to be packed up, get the finishing touches, finish its folding and any kind of modifications, and then it'll leave through one of these little vesicles, little sac, and that'll take it to the membrane where it could be exported. So there's these vesicles. It's not just this stack of pancakes, but you'll see these little balls breaking off the side. These are transport vesicles, and they can carry stuff to other organelles or to the cell membrane for export. Some of these may also store. These can become vacuoles, which store things, or lysosomes, which are these little sacs full of digestive enzymes to digest things. So there's a lot of jobs that can happen with these vesicles. So lysosomes, I mentioned, was one of these jobs. So lysosomes contain digestive enzymes. So in a microscopic organism, like a paramecium little pond organisms, this would be their digestive tract, really. I mean, they don't have a digestive tract like we do to digest their food. So they would eat their food, take it into food vacuoles, which would then go and fuse with a lysosome, and that can then break it down. But our cells have lysosomes too, even though we have a digestive system, because we may still need to break down, for example, a protein back into amino acids so we could build something new. We also may need to break down worn out cell parts, like a mitochondria that's not functioning right anymore. Or our white blood cells, remember how they fight infections. So a white blood cell might eat a bacteria, and then that would get trapped in a vesicle and go to a lysosome, and these digestive enzymes would digest it. So that would be another example of something a lysosome might do. Again, notice how these are all sort of related to one another. So they're separate organelles, but it's sort of like your heart beats, but it doesn't just beat for the heck of it, right? Your heart's beating to pump blood to other cells that need it, and your muscles contract, but they contract because your brain coordinates with them to move or do other things. So just how your organs communicate with one another, the cell organelles also are related to one another. And so the rough and smooth ER products here travel to the Golgi where they can get modified and packed. The Golgi then makes these vesicles, which notice here can export things or can become lysosomes. So these are all related to each other. All right, vacuoles in animals, not all that obvious from a picture. They look very much like a vesicle or a lysosome. They just look like a little sac. However, really, really important, if you are asked to identify whether something is a plant cell or an animal cell, this central vacuole is going to be one huge giveaway that it is a plant cell. It is bigger even than the nucleus in a lot of pictures. And there's a third vacuole called a contractile vacuole. We're not really going to spend much time on that one. But some microorganisms that live in like lakes, for example, the paramecium, if you've ever seen diagrams of a paramecium, they're little microscopic organisms. And they live in fresh water, and water can leak in. So obviously they don't want to explode, and so they have this thing called a contractile vacuole, and it can squirt out excess water. But of all the vacuoles, make sure you know the central vacuole of the plant cell. It's really, really big. And it doesn't just store water. Here's the thing. It creates pressure on the cell membrane and the cell wall. And that's what makes the plant cells stiff. That's why plants don't look wilted. If you don't water your plant, the reason they look wilted is specifically because the vacuoles, that was full of water, it loses some of its water. And so as this shrinks, the cell kind of collapses a little bit. It still has a cell wall, which is pretty sturdy, so it doesn't collapse like an animal cell would literally shrivel up into this tiny ball. But it does collapse enough that the plant looks all wilted, because there's not enough water in here to create pressure. So make sure you know that the vacuole in a plant, let me erase here so you can see it a little better. Look how big that is inside of the plant cell. And it's pushing everything else outward to the cell wall. This is a contractile vacuole in a paramecium. So it looks almost like a little flower. This is the one that squirts out excess water. All right, mitochondria are really important for energy. They take the energy in sugar and they break it down and will release it. And they make something called ATP. It's called cell respiration. We're going to talk about it. We have a whole chapter about it. But in essence, this is the powerhouse, you probably heard, of the cell. It breaks down sugar and then converts the energy that was trapped in that into something called ATP, which is sort of your cell's energy currency. It has a double membrane. Interesting side note, all the mitochondria in your body, they come from other mitochondria, obviously, when your cells reproduce, but all your mitochondria originated from your mom, never your dad. It turns out that all the mitochondria, remember, you come from an egg and a sperm. The egg has mitochondria in it. The mitochondria in the sperm are actually back here outside of where the nucleus is. They're used to power the tail so he can swim. And the only part of a sperm that actually enters the egg is the nucleus. And so therefore, all your mitochondria are inherited from your mother, because your dad's mitochondria don't get passed on. So if you're a guy, you will never pass your mitochondria to your children. Interestingly, and that's used in forensics, by the way, mitochondrial DNA can be used to trace genealogy on the maternal side, on the mother's side. It has two membranes. The inner one is very folded. It looks like this. This is the inner membrane, and then this is the outer membrane of the mitochondria. And this is what it looks like under an electron microscope. All right, and then chloroplasts, kind of similar in the fact that they also are related to energy, but they don't do cell respiration, they do photosynthesis. And they are unique to plant cells. Animal cells do not have chloroplasts. So this is what chloroplasts look like, a little bit different. And quick comment, very important for test purposes. So some people have a misconception that mitochondria are in animal cells and then chloroplasts are in plant cells. And that is not correct. The correct thing is that mitochondria are pretty much in all eukaryotic cells. So mitochondria, because all cells need energy. So no bacteria, but all eukaryotic cells, both plant and animal cells, have mitochondria. But only plant cells would have chloroplasts. So don't make that mistake and think that mitochondria are animals and chloroplasts are plants. Mitochondria are all eukaryotic cells, and then chloroplasts are plants. Okay, endosymbiotic theory. So this is a theory about where mitochondria and chloroplasts came from. And it actually has a lot of support, so it may sound a little far out, but I'll show you the evidence, and I think you'll see why they think this. So they think that mitochondria and chloroplasts were actually bacteria that lived on their own. And they were eaten by other bacteria, and they became a part of them. Why do we think this makes sense? Well, because, so if a cell eats a bacterium, like if your cells were to eat a bacteria, here's what would happen. your cell membrane would surround it, and it would make a double membrane. This would be the inner membrane would actually be the membrane that originally belonged to the bacteria, and the outer membrane would be a piece of your cell membrane. I mean, that's endocytosis. That's what happens when your cells engulf something. Well, guess what? Mitochondria and chloroplasts both have a double membrane, an inner one and an outer one, which is exactly what you would see if a cell ate another cell. Also, mitochondria and chloroplasts have their own DNA. So if they were cells living on their own, they would have to have had their own DNA. Well, they still do. And it's very much bacterial DNA. They don't have a nucleus, but they have their own DNA. They even reproduce themselves. Within your cell, they kind of control their own cell reproduction. So there's a lot of evidence that shows that mitochondria and chloroplasts, now this doesn't show the double membrane, but imagine there would actually be two membranes here, because this would become part of an outer membrane. But this is the idea. You start with two bacteria, one ate the other, one lives inside the other. Eventually, this is symbiosis, they both benefit, but eventually this one loses its ability to live on its own, and it just becomes a part of the bigger one. So that's endosymbiotic theory. Your cell, a lot of people think that the cytoplasm, everything's just floating around. That's not accurate. That would be like saying that your lungs and your heart and everything are just bouncing around, floating in your body. We know they're held in place. Well, the same is true for your cell parts. There's something called the cytoskeleton, and these are proteins that basically give the cell shape, and they also help cells move. The cells that move, like, for example, muscle cells that contract, or sperm cells that swim. So there are a couple of different parts of the cytoskeleton. One is the microfilaments. They're made of a protein called actin. These have the ability to contract. So this is what allows your muscle cells to contract. This is also what allows, like an amoeba, which looks almost like a blob, to stretch out its what are called pseudopods and move in a pond. Microtubules are bigger. They are hollow, and they are the ones that form cilia and flagella. So this is a flagellum. These would be cilia, lots of little hairs, and these would allow small organisms to swim, for example. And there's also these organelles involved in cell reproduction called centrioles. Only animal cells have them, and those are also made of microtubules. In fact, I made a little note here that only animal cells have centrioles. And I'll probably show you in another slide, but the centrioles look almost like this, like T-shaped, like that. And when the cell goes to divide in an animal cell, they end up on the edges, and then these fibers form across, and the chromosomes line up in the middle, and then the cell can divide. But these are the centrioles, and they're actually there all the time. This is kind of a picture to show you the difference between the microtubules. They're big and hollow, and you can see them here. And then the microfilaments, which are much smaller. There's also these intermediate sized ones, but we didn't really talk about those. Here's a picture of microtubules. So cilia, flagella, these are on microorganisms, but there's, also like sperm cells have a flagellum. And then this is the centrioles. This is what they look like, and when the cell goes to divide, they're kind of found here at the ends. All right, and then cell walls. Cell walls, I mentioned, they're actually found in lots of organisms, not just plants. They're also found in fungi. Bacteria have a version of a cell wall as well. They do not control what enters and leaves. That's the cell membrane. Don't mix that up. They are only for support and protection and shape. So be aware of that. It's not like, oh, plants have cell walls instead of cell membranes. No, they have both. They still have the membrane, and then also the cell wall. Notice the nucleus, here's that giant vacuole, mitochondria, and chloroplasts. And so just to summarize, this is a generalized animal cell. A lot of times they're kind of shown roundish, although they're not all round. Notice the centrioles, plant cells don't have those. And we don't see a really big vacuole or chloroplast. And now here's a plant cell. Notice how a lot of times will show it rectangular, but they're not all rectangular. No centrioles, giant vacuole. It's not a great drawing of one, but here's that big vacuole. And also we see chloroplasts, but we still see mitochondria. So that kind of gives you a summary of everything. And then I didn't show here a picture, but I believe it was at the beginning, of the bacterial cell, which is prokaryotic, no nucleus, no organelles. It would just be DNA and then the ribosomes, and that's it.